Bismillahirrahmanirrahim. Dear students, have a nice day. Hope you will be fine and best of your health. This is Dr. Sajid Mahmood Rao, Division of Science and Technology, University of Education, Lahore. Today we are talking about phytochemicals, in which we will discuss short question answers regarding to the course of natural product that is relevant to the Chem 4119. Let's see short question number 7. Write a short note on the following phytoconstituents, that is glycosides, polyketides, and tannins with their identification test. Let's see what are the glycosides. Glycosides are the phytoconstituents that has been derived from the plant sources, and glycoside is any molecule in which a sugar group is bonded through its anomeric carbon atom to another group and to form glycosidic bonds via oxygen atom or sulfur atom. So the sugar group is known as the glycone part, and the non-sugar group is generally known as the aglycone part. Basically, aglycone part is the general group of these molecules. So you can see over here in the structures of cardiac glycoside, which is the general structures of this molecule. In these structures, glycosides as a glucose moieties or substituents are attached to the basic structures of the steroid molecules. So this linkage is due to the presence of oxygen atom, and this oxygen atom is generally known as anomeric oxygen atom, and the carbon which is attached to this oxygen atom is called the anomeric carbon atom. Similarly, most of the glycosides or the glucose is also attached with the alkaloids or the terpenes molecules, and we can see those molecules as a terpenoidal glycosides or the alkaloidal glycosides. So you can see over here for the two more structures, that is salicine and glucovanillin. In salicine, the glucose moiety is attached with the methanolic groups, and in glucovanillin, the glucose moiety is attached with the benzaldehyde or methoxybenzaldehyde group. So in these two groups, the basic glucose moiety is attached with the anomeric oxygen or the anomeric carbon atoms. Salicine is isolated from willow salix bark. It is found in the bark of poplar species and the leaves of the willows and poplars. It is also found in castoreums. It is used as an analgesic agent, anti-inflammatory and antipyretic drugs. The second molecule, that is glucovanillin, it is isolated from vanilla beans, and it is used as antimicrobial, antioxidant, anti-mutagenic and anti-sickle cell disease active drugs. Let's see the solubility of glycosides. Since glycosides have two parts, one is aglycone part and the other is glycone part. Aglycone part is insoluble in water, while glycone part is water soluble. But overall glycosides are water soluble and insoluble in organic solvents. Let's see the identification test of glycosides. Glycosides can be identified by two tests, that are Lieberman-Butcher test, and the other is Kalyani test. So in the first test, we take 2 ml of extract and add 2 ml of acetic acid and 2 ml of chloroform, then cool the solutions and then added concentrated sulfuric acid. In the results, green color showed the entity of a glycone, that is the steroidal part of glycosides. So in this way, the glycosides are indicated. The second test is Kalyani test. In this test, a solution of glacial acetic acid, that is 4 ml, with the 1 drop of 2% trichloride mixture was mixed with the 10 ml of aqueous plant extract and 1 ml of concentrated sulfuric acid is added. In the results, a brown ring formed between the junctions of the two layers, which indicates the presence of cardiac steroidal glycosides. When we see the sources of glycosides, these are isolated from Digitalis family, Senna leaves and Senna pods and beer berry. The biological activity, when we see, it behaves as cardiotonic agent, which is due to the presence of Digitalis glycosides, and it behaves as laxatives due to the presence of anthraquinone glycosides, and it works as anti-inflammatory agent due to the presence of glycyrrhizine glycosides. So many other phytoconstituents are also present within the glycosides, and that phytoconstituents are rutin, quercetin, and hesperidine. Glycosides are also used for the treatment of heart failure, as well as it is used as novel anti-neoplastic agents. A large range of antibiotics are prepared from glycosides. So now we can see some important pharmaceutical brands which have been launched to the market on the basis of different phytoconstituents that fall in the category of glycosides. So the first brand is Bioflavonoids. It is manufactured by Douglas Laboratories and we take its dose to 50 milligrams orally every 12 hours or 3 times a day. It is formulated in tablet dosage form. It is used to enhance the actions of vitamin C, to support blood circulations as an antioxidant, and to treat allergies, virus or arthritis and other inflammatory conditions. The second brand is Gingko Biloba Certified Extract. It is manufactured by Life Extension Laboratories and we take its dose as 1 capsule per day. It is formulated in the capsule dosage form and it is used to support brain health and memory and circulations. This is another brand that have been launched by the Ali Nutraceuticals and the name of this brand is Black Osh. We take its dose 1 capsule per day and it is formulated in capsule dosage form. It is rich with pharmaceutical applications and medicinal applications because it is used a large number of diseases that includes menopause, sleep, headache, hot flashes, mood changes, sleep problems, heart palpitations, night sweats, as well as vaginal dryness. Let's see what are the polyketides. Polyketides are a large group of metabolites found in the bacteria, fungi, and plants which are synthesized from acyl coenzyme precursors. So, polyketides are a diverse class of compounds that are often created by a series of molecular enzymes which either contain alternating carbonyl or methyl groups, that is in the form of C double bond O and CH2. So you can see over here two examples of the phytoconstituents that fall in the category of polyketides. And these two phytoconstituents are natamycin and gallic. Now let's see what is the natamycin. Natamycin, also known as pimaricin, is an antifungal medication used to treat fungal infections around the eye. This includes infections of the eyelids, conjunctiva and cornea. It is also used as eye drops. Natamycin is also used in the food industry as a preservative. However, these have some allergic reactions by its using as an oral dosage form. So, the second molecule is gladamycin. Let's see what is the functions and the uses of the gladamycin. Gladamycin is a 1,4-benzoquinone annamycin antitumor antibiotic that inhibits the functions of HSP90 by binding to the unusual ADP and ATP binding pocket of the proteins. HSP90 client proteins play important roles in the regulations of the cell cycle, cell growth, cell survival, apoptosis, angiogenesis, and oncogenesis. Polyketides are insoluble in water. However, these are soluble in organic solvents, and that solvents are chloroform, methanol, and dimethyl sulfur oxides. Now we see the identification test through which how can we observe and confirm the presence of polyketides. In the first test, we take the sample, that is 5 mg, and add 2 ml of sulfuric acid and shake gently. If reddish brown color produced, then it means the erythromycin is present, that is the class of polyketides. In the second test, we take 5 mg of sample extract and added 5 ml of xanthodiol and heat on water bath. If red color produced, then it means polyketides are present. Polyketides are isolated from different sources. The main sources from which polyketides are isolated and extracted are for bacteria, fungi, plants, certain marine animals. Polyketides also have some biological activities, and these work as antibiotics, antifungal, cancer chemotherapeutics, and antiparasitic effect. So you can see over here some important phytoconstituents, and these phytoconstituents are macroleads, annamycins, polyenes, picromycin, gallic, and natamycin. Now we discuss its uses and importance turn by turn. So the first molecule is macroleads. Macroleads are a class of natural products that consist of a large macrocyclic lactone ring to which one or more deoxygenated sugars, usually cladinose and D-ososomines, may be attached. The electron rings are usually 14, 15, and 16 membered. Macroleads belong to the polyketide class of natural products. The second molecule is enzymycines. Enzymycines is a family of bacterial secondary metaboloids that show antimicrobial activity against many gram-positive and gram-negative bacteria and includes various compounds including streptovaricines and rifamycins. The next molecule is picromycines. Picromycin was studied by Brockman and Haeckel in 1951 and was the first antibiotic macrolide to be isolated. Picromycin is synthesized through a type 1 polyketide synthase. Sister means Streptomyces venezuela, a species of gram-positive bacterium in the genus Streptomyces. The next two phytoconstituents that are gliadinamycins and atomycin have been discussed previously. Polyketides also have some applications. These are also used as immune system suppressant. They are also used as cholesterol lowering agents. Now we discuss some important pharmaceutical brands which have been launched on the basis of phytoconstituents that fall in the category of polyketides. So the name of first brand is Erythrocene. It is manufactured by Pfizer Nutraceuticals and we take its dose 500 mg per day. This brand has been formulated in tablet dosage form and it is used in pneumonia conditions. It is used as ear infections and bronchitis. The next molecule is amphotericin B, which is manufactured by Bioflavonoids Nutraceuticals and we take its dose 50 mg per day. It is formulated in injection dosage form and it works as antifungal agents. Let's see what are the tannins. Tannins are a class of polyphenolic bimolecules that bind to and precipitate proteins and various other organic compounds, including amino acids and alkaloids. They mainly contain more than 5 hydroxyl groups. In addition to tanning leather, tannins are used in photography as mordants in dyeing, clarifying wine and beer by precipitating proteins out of them. And as astringent in medicines, tannins are found commonly in the bark of trees, wood, leaves, bird stems, fruits, seeds, roots, and plant galls. So, the tannins, also called tannic acid, any of a group of pale yellow to light brown amorphous substances in the form of powder, flakes, or a spongy mass, widely distributed in the plants used chiefly in tanning leather, dyeing fabric, making ink, and in various medical applications. So you can see over here two further important examples that fall in the category of tannins, and these are the phytoconstituents that are gallic acid and the basic skeleton of tannins, in which different polyhydroxy groups are condensed with each other and known as general structures of the condensed tannins. So let's see the sources and uses of gallic acid. Gallic acid is trihydroxybenzoic acid, a type of uric acid found in gallnuts, sumac, witch hazel, tea leaves, oak bark, and other plants. The chemical formula of gallic acid is C6H2OH3COOH. It is found both free and as a part of hydrolyzable tannins. So gallic acid is used as antioxidants and helps to protect our cells against oxidative damage. Gallic acid was found to show cytotoxicity against cancer cells without harming healthy cells. Gallic acid is used as a remote astringent in cases of internal hemorrhage. Gallic acid is also used for the treatment of diabetes mellitus. Due to the presence of hydroxyl group in the basic skeleton of the tannin molecules, these are easily soluble in water as well as in organic solvents. The organic solvents which are used for the solubility of tannin molecules are methanol, ethanol, butanol, ethyl acetate, and chloroform. Now we see the identification test. This test is carried out by using gold beater skin test. It is carried out when a small piece of gold beater skin membrane prepared from the intestine of an ox is soaked in 20% hydrochloric acid, rinsed with distilled water, and placed in a solution of tannin for 5 minutes. A brown or black color is produced on the skin due to the presence of tannins, which indicates the presence of tannin molecules. The second test is Swinozone test. This test is carried out when a mixture of aqueous extract of a drug and sodium acid phosphate is heated and cooled and filtrated. A solution of phenazone is added to the filtrate. A bulky colored precipitate is formed, which indicates the presence of tannin molecules. Tannins are found in most part of the plants, and these plants are Potentilla erecta, in which tannins are found in the roots of these plants. Second plant is Cesium Cumini. In this plant, the tannins are found in the bark of the plants. The next plant is Acacia Nylotica. In this plant, the tannins are found in the pod of this plant. Tin is also active agents, which is due to the presence of Pontilier acta. In this plant, it is have antiviral, anti-inflammatory activity. The second plant, Chlamydia trianda. In this plant, antibacterial and antidiarrheal activity is found. The next plant is Cesium Cumini. Anti-asthma and anti-ulcer activity is found in this plant. So, we discuss some important phytoconstituents which are found in these plants, and these phytoconstituents are tannic acid, pyrimidic acid, and ellagitannins. So here we discuss some important phytoconstituents, their uses, and isolation sources. The first phytoconstituent is tannic acid. Tannic acid is found in the seeds, bark, cones, and heartwood plants. Tannic acid is a common mordant used in the dyeing process for cellulose fibers such as cotton, often combined with alum and iron. Nowadays, tannic acid is applied typically for the treatment of cold sores, diaper rashes, fever blisters, and poison ivy. Tannic acid is also taken by mouth as oral drug and applied directly for bleeding, chronic diarrhea, dysentery, bloody urine, painful joints, as well as persistent cough and cancer. The second molecule, that is ellagitannins. Ellagitannins are used and isolated from as these are used as bioactive polyphenols that are found in some fruits, nuts, and seeds such as pomegranates, black raspberries, strawberries, walnuts, and almonds. These are used as a universal phytoconstituents. The universal nature of these mechanisms in common forms of cancer suggests that pomegranates ellagitannins, which have been tested in both prostate and colon cancer cells by our research groups, and also be useful dietary agents for the prevention and treatment of other forms of cancer such as breast cancer. So tannins are also used in tanning industries, used to produce different colors in ink formations, as well as used in pharmaceutical sectors. Now we discuss some important pharmaceutical brands that have been launched on the basis of phytoconstituents that fall in the category of tannins. The first brand is Erythrocene. It is manufactured by Vitamin Company Nutraceuticals, and we take its dose 2 to 3 tablets per day. It is formulated in tablet dosage form. It is used in obesity, means it is used in weight loss. The second brand is Pasphone. It is manufactured by High Mount Pharmaceuticals, and it is taken 6 tablets per day. It is formulated in tablet dosage form, and it is used as antispasmodic agents. Dear student, thank you so much for your kind attention. So please feel free to contact in case of any queries. Okay, Allah Hafiz, bye.